Hello fellow botanists, welcome to our next plant lecture, and this lecture we're going to look at our first major group of plants. We're going to look at the non-vascular plants. So as a reminder of where we are taxonomically, we're talking about plants which are embryophytes. So these are plants in which the gametophyte nurtures a little developing sporophyte. And we saw this on our tree number 2, or origin of plants. And in particular, in this lecture, we're going to look at non-vascular plants. And so non-vascular plants can go by a few names. Non-vascular is one of them. They're also referred to as bryophytes with an ES, but be careful if you say bryophyta, you're referring to a specific phylum of plant within this group. So on our tree number 2, we see our bryophytes, our non-vascular plants right here. And they're separate, of course, from other vascular plants that we'll get to in our next lectures. And you also see our bryophytes over here on the phylogenetic tree from your textbook. And remember, of course, that this tree is just oriented horizontally instead of vertically. So I'll move over here so we can see where we're going in this lecture. And I'm going to talk about 3 things generally. The first is I'm going to talk about our general characteristics of non-vascular plants. We're going to think about tissues, about organs, and surprise, they don't have any. And we're going to think about variations of the life cycle. Then I'll take a look at the 3 major phyla. So remember, this is the taxonomic unit just below kingdom, the plant kingdom in this case. I'm going to take a look at our 3 major phyla, our Hepatophyta, which are the liverworts, our Anthocerophyta, which are the hornworts, and our Bryophyta, which are the mosses. And finally, I'd like to end by thinking about the ecological importance of this particular group, because as always, these groups are far more than just a bunch of names on a phylogenetic tree or just a bunch of morphology. They play some really important roles in our world. So I'd like to mention those. So a quick reminder about plants, and remember that all plants have a few features in common. And so one of the features is they have a life cycle with an alternation of generations, alternating between the 1N gametophyte, the plant that produces gametes, which are held in Antheridia and Archegonia, and the 2N sporophyte generation. And so the sporophyte is the plant that produces spores. The spores are held in a sporangium, and the spores are produced by meiosis to complete our life cycle. In addition to this alternation of generations, because there are some non-plants out there, a bunch of algae that do this, what's particularly unique in the plant life cycle is that the gametophyte nurtures the embryo sporophyte. So the zygote, remember, is not fertilized by a sperm and an egg. They're sort of free swimming. The egg is actually held onto by the gametophyte. The sperm fertilizes that egg. Then the zygote is held onto by the gametophyte, and as it starts to divide, this dividing sporophyte is nurtured by that gametophyte. All right, so it's the embryo sporophyte. And finally, remember that plants also have apical meristems. And so this is sort of like the bud of a plant, if you want to think about it like that. It's a region of undifferentiated cells that keeps dividing and dividing and dividing to produce more tissue for the plant. And we'll talk more about apical meristems in some lectures on plant development, but for the moment, just note the apical meristem is going to help our plant grow in some very particular directions, and of course give rise to particular structures that we see in our groups of plants. All right, so if plants have all of these features in common, that makes them a monophyletic group. If you remember our discussion of the term monophylogeny from our lab earlier. However, the first group of plants that we're going to look at, our non-vascular plants, are paraphyletic. And so the idea behind a paraphyletic group, again, as you may remember, is that this group has a common ancestor, but there are some other plants that are that share this common ancestor but are excluded from the group. So as we see in this particular tree, here's a common ancestor that our 3 major phyla of bryophytes have, but this common ancestor also is the common ancestor for other plants, and we're excluding these in our group of bryophytes. So that makes bryophytes a paraphyletic group. And we see the same paraphyletic grouping over here in this phylogenetic tree from your textbook. All right? 3 phyla over here, a common ancestor, but we're excluding all of these other plants that also have the same common ancestors. And so because non-vascular plants are a paraphyletic group, there aren't a common set of features that all of them happen to have. So we'll talk generally about some features that some bryophytes can have, but notice that there's sort of nothing that uniquely defines a bryophyte from all other plants. All right, so what can we say about our bryophytes, our non-vascular plants? Well, unsurprisingly, as the name implies, they don't have any vascular tissue. And because non-vascular plants don't have any vascular tissue, how do they get what they need? And there are a couple of ways that they can do this. So one way, of course, is they can just absorb what they need directly from the environment. So this is a little liverwort right here, and you can see that it's very flat and close to the ground. And so it's able to absorb most of what it needs in terms of water and minerals directly from the ground into its individual cells. All right, now, some non-vascular plants are a little bit taller and a little bit bigger than our cute little liverwort over here. So if we look at our mosses, they're a bit taller, and clearly if you are going to be a moss gametophyte, you do have to move some compounds around. You have to move water, you have to move minerals, you have to move carbohydrates. But we don't have vascular tissue. Instead, we have structures that are analogous to xylem and phloem. We have hydroids, and as the hyd part implies, they move water, sort of like xylem. However, it's not xylem because these hydroid cells lack lignin, that very particular compound that strengthens the secondary walls of xylem cells. And then we have leptoids, which are analogous to phloem, and they move carbohydrates. All right, so if we don't have vascular tissue, what that means is that we also don't have any true organs. So we have no true leaves, no true stems, and no true roots. Instead, we have a few other terms for talking about structures that are kind of leaf-like or kind of root-like. And so we talk about the bodies of non-vascular plants as being thalloid or leafy. And so a thalloid body is sort of a very undifferentiated blobby-like body, as you see with this liverwort over here. Not ugly, just different. Our mosses, though, can have more of a leafy sort of body, and you can see some leaf-like structures over here. So remember that these aren't true leaves, they don't have vascular tissue, but of course they look like leaves, and functionally they're sort of analogous to leaves. And we also have some structures that aren't roots, but are functionally analogous to roots in that they help to anchor the plant, and those are rhizoids. And I don't have a good rhizoid picture here for either of our non-vascular plants, but you'll get the chance to see these in lab. Our third feature, of course, is the life cycle. And remember that all plants have the same general life cycle. So here's a quick reminder of what it is. So we have two parts. We have a 2N generation, we have a 1N generation. And so if we have a gametophyte over here, which I've shown as sort of a general amorphous sort of gametophyte, like one of our liverworts over here, it can produce gametes, it can produce sperm cells in an antheridium, which is the male gametangium or the male gamete container. It can produce egg cells in an archegonium or the female gametangium, the female gamete container. And then if a sperm cell fertilizes an egg that's held onto in the archegonium over here, we get fertilization. And so the zygote over here is now 2N, and I've put it above our 2N line, even though in reality it hasn't left the archegonium over here. So our zygote is now a 2N cell, and it divides by mitosis a whole bunch of times to produce our sporophyte. And our sporophyte can have a sporangium or a spore container. And of course, inside this, we have lots of spores that are produced by meiosis. So meiosis occurs, which takes us back into the realm of 1N cells, and each meiotic division gives us 4 cells. So I've drawn 4 spores over here. And then if we take one of these spores, mitosis, it can divide by mitosis to give us this gametophyte again. All right, so that's the picture of the general life cycle, but let's actually connect this now to moss plants that we see, and we'll see if we can identify these features over here in moss plants. So let's start over here with the gametophyte. In our moss diagram over here, there are actually 2 gametophytes. All right, so this is a leafy green gametophyte, this is a leafy green gametophyte. And this gametophyte at its very tip over here is going to have a gametangium. Specifically, it's going to have an antheridium. And our antheridium, of course, is our sperm container. And you can see lots of sperm cells. On this branch of the gametophyte over here at the very top, we have our other gamete container. We have the archegonium. And our archegonium is our egg container. Now, if a drop of water happens to land fortuitously on the antheridium, it can open and release the sperm cells into this drop of water, which then hopefully falls over here onto the top of our archegonium, which is this long, it has a very long neck. And at the base of the archegonium over here is our egg cell. Now, if a sperm cell can swim down the long neck and fertilize the egg cell, we get a zygote. So notice that fertilization has happened. This box indicates fertilization over here. We've now gone from a 1N part of the life cycle to the 2N part of the life cycle. So this is actually a reverse of the way I've ordered it over here. They'll flip the life cycles back and forth, but I'll always try to stay with 2N on the top. So we have we're in the 2N part of the life cycle, and our zygote is going to start dividing by mitosis. And so here we see our little embryo sporophyte. It's starting to grow, and eventually our embryo sporophyte turns into a full-fledged sporophyte. So this whole brown thing here is our sporophyte. And at the tip of our sporophyte, this little structure right here, is the sporangium. It has lots of spores that were produced by meiosis. So this box over here indicates that meiosis has occurred. We're now back in 1N territory. We have a bunch of spores. So these would be spores, and if our spores start to divide by mitosis, as they are over here, eventually we return to our gametophyte, our multicellular gametophyte. It's sort of the key takeaway from all of this. And one of the key points that I want you to hold on to this is that the gametophyte generation is dominant. The sporophyte is actually growing out of the gametophyte. So we see this in a couple of pictures here. In the top cartoon picture, our orange generation over here is the gametophyte, and our purple generation is the sporophyte. Right? Now we actually see these in a real picture, of course, green gametophyte, sporophyte, and we can see that the sporophyte is dependent on the gametophyte. In fact, so dependent that often it's a brown color and doesn't even do a lot of its own photosynthesis. As we take a look at other groups of plants, our seedless plants and eventually our seed plants, we're going to see that this pattern changes. So we're going to expand both series of pictures in subsequent lectures, and you're going to see how this pattern shifts for other major groups of plants. So what are our major phyla of non-vascular plants? And we have three of them, as I said. The first one are liverworts, or hepatophyta. So phyte, of course, denotes a phylum over here of plants, and hep often denotes liver. And so if you've studied animal biology at all, often any functions associated with the liver have a hep root in them somewhere. All right, so the gametophytes of our liverworts tend to be very primitive, and in fact liverworts are thought of as being having some of the most ancestral or primitive characteristics of our three non-vascular plant phyla, although they do, as we'll see in a second, have some very interesting structures that other groups don't have. So they're considered primitive because they're small, and in fact they're often just sort of very flat. As you can see in both of these pictures, they grow right along the ground, and they can absorb everything that they need in terms of water and minerals so that they don't have conducting cells. And they also don't have stomata, again those openings that can be regulated by guard cells. Instead, they have pores, which are these openings right through the cuticle that allow for gas exchange. All right. Now, what they do have, though, that's really interesting is they have gametophores. And so here's a new term, but we can use our understanding of language to figure out what it means. So first, remember that for a new word, a botanical word, we're going to read it right to left, and we're going to start with the for root over here. So for means to bear. And you have probably heard of some forewords before. You might not have realized it, but one of the more common forewords you might have heard is the male name Christophor, which means literally a bearer of Christ. All right, so in this case, we're going to have a for that bears gametangia, or structures that hold gametes. And so we're going to have two flavors of gametophores. We're going to have an archegoniophore, which bears archegonia, and an antheridiophore that bears antheridia. So our diagram over here indicates or shows each of these gametophores. Here is the female gametophore over here, or archegoniophore. It's this little stalk over here with these things that look like little palm fronds. And underneath the palm fronds are the archegonia. And so here would be a little archegonium. It would have an egg inside it. And once this egg is fertilized and becomes a zygote and starts to divide, this structure right here is the whole, basically, of the sporophyte. So as you can see, the sporophyte is really small over here. And we'll formally mention that in just a second. All right, so that's the archegoniophore. You can see the little sporophytes dangling from below it. And the antheridiophore looks a little bit different. So this is an antheridiophore. So the antheridiophores from above look sort of like umbrellas, whereas the archegoniophores look sort of more like little palm fronds, or maybe like just the spokes of the umbrella if a really bad windstorm has pulled away all of the lining of the umbrella. So a final note, or a word about sporophytes. As with all non-vascular plants, the sporophytes are dependent on the gametophyte, and they are very small, as we mentioned, even by the standards of moss. They're just these tiny little structures that are dangling down from one of the archegoniophores. Our second group of non-vascular plants are the Anthocerophyta, or hornworts. All right, so Anthocera, as you might guess, means horn. And so their gametophytes are primitive, somewhat like the gametophytes of liverworts. And so they tend to be flat. You can see in this picture they're not very tall at all. They're fairly flat, and so they can absorb basically what they need from directly into their cells from the ground. We're really interested in the anthocerophytes, though, for what their sporophytes do. And the sporophytes are still dependent. That hasn't changed. But what has changed is that they are less small. They're a little bit taller relative to liverworts. So this whole structure right here is a sporophyte. Whole thing right here. And so much bigger than the tiny little sporophytes we saw for liverworts. And the reason that it can be taller is that it has stomata, not the gametophyte, but the sporophyte. So as the openings of the as the guard cells of the stomata open and close, it regulates water loss. And so if the stomata is open, then water can evaporate, creating a little bit of a vacuum and pulling up water from further below. All right, so this is sort of like what happens in the xylem cells of plants, but of course we don't have xylem, so this is just sort of a smaller version of it, but we can sort of use a little bit of a negative pressure gradient to create a straw that draws water up. All right, so that's why our sporophytes of the hornworts can start to be a little bit taller. And now we get to our third group of non-vascular plants, the Bryophyta, or mosses. And so the gametophytes are certainly a little bit more derived. They have some features that the gametophytes of our liverworts and our hornworts didn't have. And so they're taller. And so clearly they're not just flat, they're somewhat taller. And what makes it possible for them to be taller is that they have conducting cells. So not xylem, not phloem, but hydroids and leptoids. And so remember, hydroids are the cells that move water, leptoids are the cells that move carbohydrates, and they're not xylem and phloem, they lack lignin, but they can move and circulate carbohydrates and water and minerals and nutrients around the plant body. All right? And the gametophytes themselves have pores, not stomata, but when we think about the sporophytes again, like our hornworts, have stomata. In other words, our hornworts were the first plants to have stomata, and all subsequent plants have stomata as well. Which means that the sporophytes of our mosses can also be a bit taller, and they can do some really neat things like disperse spores. So I'll move over here so you can see the spore dispersal happening. And so this is a little sporophyte of sphagnum moss. So the gametophyte's down here. This is the sporophyte. At the top of the sporophyte we have a sporangium. At the very top of the sporangium we have a little bit of a lid almost. And so sphagnum moss does this better than a lot of other moss species, but there are varieties of this in a lot of moss species. And so what happens is, as the sporangium matures, it actually shrinks. And as it shrinks, there's gas inside it that becomes it gets put under higher and higher pressure. So imagine that you have a little bag of potato chips, and you haven't opened the bag, but you crunch up all the potato chips inside, trimming the potato chip crumbs. And then you start to squeeze that bag of potato chips. And eventually, if you squeeze that bag long enough, it's going to pop open and shoot potato chips all over the place, right? Particularly if you sort of pop it all at once, right? Not that any of you ever did that as kids or bigger kids, right? But this is the way that the sporangia of mosses distribute spores. And in this picture over here, you can see this exploding capsule. So here is the capsule just before the lid pops off, and the lid pops off and shoots out these spores really at really a sort of explosive, astonishing speeds, pretty close to 90 miles an hour initially as they're emerging from the sporangium. So this is almost the speed of a fastball thrown by a major league pitcher. So quite an impressive spore dispersal mechanism that they have. I'd like to end, as I mentioned, by talking a little bit about the interesting ecological importance of our non-vascular plants. And there could be a whole lecture on the ecological importance of non-vascular plants, but I'm going to mention just a few moss species. So a lot of mosses and non-vascular plants in particular, but especially mosses, are early successional species. They can grow where no other plants are growing. So on rock faces, even on the sides of buildings or the sides of houses, if you ever pressure wash the side of a house. And then once our non-vascular plants start growing, they can provide the habitat for other seeds to start growing as well, and sort of really getting the vegetative ecosystem going on a very rocky surface or a very sandy surface that doesn't have any existing vegetation. In fact, there can even be little moss, they're called little moss mice, that roll across glaciers and start colonizing glaciers. All right. In fact, mosses are so good at early succession that one species of moss, or one genus of moss in particular, sphagnum, can colonize open water, especially the open water of bogs. And so bogs are sort of these little very shallow water ecosystems over here that don't have great drainage and also aren't very disturbed. So over time, sphagnum will grow over the surfaces of bogs and sort of create what's like the surface of a giant waterbed. So if you liked walking across a waterbed as a kid, now imagine doing this as an adult, but the waterbed is much bigger, right? That's what it's sort of like to walk on a sphagnum bog. It's a lot of fun. If you ever get the chance to do it, do it. Right? But sphagnum bogs have a really important ecological role as well, and they are carbon sinks. So sphagnum bogs cover about 3% of the land area of Earth, but they contain 10 times that amount of carbon, like 20 to 30% of the carbon on the planet. So they're very important at trapping carbon from the atmosphere, and here's how they can do it. So sphagnum bogs tend to be very, very acidic. And so as the sphagnum grows and eventually dies, it doesn't decompose. And so the sphagnum captures carbon from the atmosphere and then basically stores it in the dead plant matter of these bogs over here. And so eventually what happens is a sphagnum bog completely fills in and becomes solid, as we see in this picture down here. And so this is of concern or interest and concern ecologically, because as our climate is warming, unfortunately, what that means is we're speeding up the decomposition rates of these peat bogs, these sphagnum bogs, which is starting to release a bit more of the carbon dioxide in these areas. And so there are a lot of peat bogs, particularly in the Arctic, which is warming faster than other parts of the world. And of course, another problem is that humans also cut into the peat bog. You often have heard about people burning peat or using peat to make peat bricks, particularly in places like Ireland and Scotland. And so as peat bogs have been destroyed by human activity, again, this releases a whole lot of carbon back into our atmosphere and destroys these very important carbon sinks that we have on our planet. So that's an ecological note. Finally, sort of a fun fact about bogs is sphagnum bogs, is they're very, very acidic, as we said. And so not only do they preserve peat, they preserve anything else that might get tossed in, such as a human body. And so there are a number of bogs, particularly in Scandinavia, where people have found dead bodies that seem to be several thousand years old. So one of the first bodies that was found was Tolland Man in Denmark over here, and you can see how well preserved the features are of this particular person, even though they're 2,000 years old. Because the bogs are so acidic, there's very little decomposition, and so you can basically get a well-preserved mummified body. And so these bodies have actually told us a lot about Bronze Age culture. And finally, one more note about the ecological importance of mosses especially, and that is that they can be great bioindicators. So mosses grow very slowly, and they tend to hang on to their vegetative tissue. And because they grow so slowly and because they absorb a lot of their water from the air around them, they tend to pick up a lot of heavy metals or other toxins in the atmosphere. And so they can give us a good picture of what's happening to the concentration of major toxins over an extended period of time. So here's an example of lead in Finland. So this is a map of Finland over here. And in this particular study, they sampled mosses of different ages from the mid-80s through the 2000s. And what you can see in the mid-80s is that there were fairly high concentrations of lead in moss plants that then eventually decreased into the 2000s. And that's because there was a lot of leaded gasoline being burned in the 1970s. But as leaded gasoline was outlawed and our atmosphere was cleaned up, the concentration of lead in our atmosphere decreased quite a bit, which is shown in the mosses, and good, of course, not just for mosses, but also us as well. We don't want lead in our air and in our drinking water, and certainly not in our own bodies, especially as we're developing children. We don't want lead interfering with our nervous system. All right, so this is our trip through our non-vascular plants. Hope you enjoyed it, and stay tuned for our next lecture on the vascular plants.